I love this night. This is a great night. Every year I really look forward to this lecture because it's very meaningful to all of us here. I'm Barry Kohler, and I'm Vice President for Medical Affairs and Physician-in-Chief of the Rockefeller University Hospital. And it's my great pleasure to welcome you all to this year's Beatrice Renfield Lecture in Research Nursing. And it's featuring Dr. Christine Miaskowski, and so we're especially pleased tonight to have her grace our shores. The university's research hospital opened its doors in October of 1910. It was the first hospital in the United States dedicated to the concept of not only treating illness, but actually finding out the causes behind the diseases. Thus, the hospital is really the birthplace of clinical research in the country. And it's also the birthplace of innovations in nursing practice, with the first director of nursing in the center there, Nancy Ellicott, introducing hospital beds on wheels, wall screens between patients, the ones that move back and forth, a rolling soiled linen carrier rather than the nurses having to go back and forth to the front of with all of the schmutz on their clothes, and as well as heated fooding carts on wheels for delivering meals. But tonight, I've never shown the next two slides before, so we'll see how this goes down. But the more you know about Miss Ellicott, the more admiration I have for her. So here's a story from the New York Times from 1909, just before she came here. Miss Nancy P. Ellicott to be superintendent of new Rockefeller Institution. Miss Nancy P. Ellicott, sister-in-law of Francis White. God knows why that's important. A well-known Baltimore citizen will go to New York on Sunday to become superintendent of the new Rockefeller Hospital. But this is only the beginning of the story. The punchline is on the next slide, which I will read to you. Miss Ellicott was for 2 years superintendent of nurses at the Church Home and Infirmary in Baltimore. She resigned over a clash of authority with the superintendent. But when that official also resigned a short time later, was nominated to succeed him. A number of the directors were opposed to the appointment of a woman to so responsible a position. But backed by, now I'm underscoring this, William Welch of Johns Hopkins and Dr. William Osler, who chanced to be in Baltimore at the time she was given the position. However, the opposition was too strong for her, and last August she resigned, tired, she said, of fighting men. I thought this would go over well in this crowd. Well, I think by the time she got to Rockefeller, she no longer had to fight men because she stayed here for the next, I think, 25 or 30 years in her position and was really a staple of the institution. I thought I would show you that first, but because I don't want to take up more time from our speaker tonight, I will restrain myself from spending the hour telling you about Miss Ellicott. But I will want to say this, and that is that the reason that we champion nurses here, and especially the research nurse, is because of this tripartite role, which I think is absolutely the centerpiece of conducting high-quality clinical investigation, because the research nurse is both the patient protector and advocate, the compassionate and expert caregiver, and also the leader or member of the scientific research team. Those three elements are absolutely central to our mission, and that's why we really believe the importance of research nursing. Let me tell you about the Beatrice Renfield and the lectureship. This lectureship honors the late Beatrice Renfield, who was a great friend to the Rockefeller University and to the nursing profession more broadly. And she really understood that investments to strengthen nursing yields benefits for patient care. And she was also aware that excellence in nursing is an essential foundation for quality health care all around. I'm delighted that Deb Lifton is here with us tonight, who joined the Rockefeller community 2 years ago, along with the president, Rick Lifton. And so we're really thrilled that you're here with us tonight. Now, I also want to recognize the Heilbronn Family Center for Research Nursing, which also is a sponsor tonight. And the Heilbronn Center is dedicated to the development of programs and collaborations to promote clinical research nursing as a unique specialty on a local, national, and also the international level. We are happy that Dr. Marilyn DeLuca, who's a representative of the Heilbronn family, but more importantly, a senior advisor to the Heilbronn Center and to me, as well as Candy, is also here tonight. So I want to recognize you as well. We also want to recognize the volunteers from NYU's Lead Honors Undergraduate Nursing Program. Any of you around? We have to, they're outside, they're volunteering. Okay. We shouldn't keep them quite so busy. At any rate, you can learn more about the Heilbronn Family Research Center by looking on our website. And last but not least, I want to thank Clinical Directors Network for webcasting this evening's lecture, and to welcome everyone who's joining us online. CDN has gained approval for educational credit by the American Academy of Family Physicians and the American Nurses Credentialing Center for attending this lecture. And finally, I'm thrilled to introduce Dr. Candy Capilli to the podium. And she joined us as the new director of the Heilbronn Family Center for Research Nursing in just last September. And so she's really been off to a fantastic start. And she's going to have the pleasure of introducing tonight's distinguished speaker.
Thanks, everyone, for coming out tonight, and thank you, Dr. Kohler. I know that Barbara is funny, but you're also, I give you thumbs up. A very nice talk, thank you. So I'd like to introduce Dr. Christine Miaskowski. She is the professor and vice chair for research in the Department of Physiological Nursing, and she's also the Sharon Lamb Endowed Chair in Symptom Management Research in the School of Nursing at UCSF. She is also the first nurse to be awarded the American Cancer Society for Clinical Research Professorship. So it's a mouthful, so I have to read because you know, she's done so much. Yeah. So Dr. Miaskowski is an internationally recognized expert in pain and symptom management. For the past 30 years, she has received continuous funding from the NIH. Her program of research focuses on determining which phenotypic and genotypic characteristics predict patients with the most severe symptoms, evaluating the harmful effects of unrelieved symptoms on patient outcomes, and developing and testing interventions to decrease symptoms in patients with cancer. Very important work that you do, Chris. So tonight, Chris will talk about benefits associated with precision health initiatives in the context of managing symptoms in cancer patients and survivors. She will also discuss strategies that can be used to phenotype cancer patients for symptom management studies. She will present data on the phenotypic and genomic characteristics that place oncology patients at risk for a higher symptom burden. The presentation will conclude with recommendations for future research and implications for clinical practice. So it is my distinct pleasure and honor to introduce this year's Beatrice Renfield Lecturer in Research Nursing, Dr. Christine Miaskowski, an accomplished nurse scientist, educator, and a cool person.
Thank you. Good evening. It's really a pleasure to be here. The mic's on, right? So this year, I celebrate my 45th anniversary as a nurse. And it's pretty neat. And I realized yesterday, 30 years ago, I defended my PhD here in New York. So it's really great to be home, all right? I'm really, really honored to give this talk because I think it's a critical issue in nursing research as well as in the arena of symptom science. And I want to begin by acknowledging how deeply honored I am to have received this lectureship. Candy was nice enough to send me some background material on Beatrice Renfield. And I just extracted from the material some of the things that touched my heart in terms of this woman. And I had the pleasure, our current dean was dean at Yale University, where Beatrice gave some of her endowment. And Kathy said that she was a superb woman. My only regret is I never got to meet her, because she must have been an amazing person to have these accolades written about her. And this whole notion that this individual, I really like the last bullet, provided strong support for research that would make a difference in patients' lives. And I'll come back to that point a little bit later in this talk. I'm hoping you're going to see that that's one of the things that drives the research we're doing at UCSF. So what would I like to do tonight? For those of you that don't do symptom research, I want to give you a little bit of a context for it in the context of the patients that I work with, who are people who have a cancer diagnosis or are survivors of cancer. And I want to put it in the context of precision health and where I see that evolving. But I really, really want to talk about inter-individual variability, because that, to me, is the key to getting to the individual patient and how we're beginning to think about and studying it. I'm going to talk about some of the work we've done, and I'm going to use fatigue as an exemplar and talk about some of the work we've done trying to understand fatigue as a single symptom. But more importantly, I want to talk about multiple co-occurring symptoms. And I'm hoping we're going to be able to have a dialogue about it, because the data I'm going to show you, I believe, happens across chronic medical conditions. So you're going to either prove me wrong or prove me right in thinking about that. And if we have time, because I know we started a little bit later, I'm going to talk to you a little bit about some work we're doing related to symptom clusters, where we're collaborating with a group at the University of Surrey trying to use network analysis, which I am really, really excited about. So if we get short of time, I'll show you a few pictures to show where we're going with that. And then our group has been really, really interested in trying to understand genomic markers and how they help us sort and predict symptoms. So I'm going to give you a whirlwind tour of some of the work that we're doing. So I think precision medicine, as it was originally called, was really about trying to understand how to treat disease better. And it was really about questions like this. People have a single diagnosis, let's say breast cancer, and the drugs don't work for everybody. We know that. So can we take different approaches to trying to sort that question and understand this inter-individual variability? The field, in my view, has moved exceedingly fast. All right, and the future is really bright. I wish I was like 40 years younger and we're starting my research career, because I think we're moving truly into the notion of precision health. And the amount of data that is going to come to patients, to providers, to researchers from a variety of sources are going to explode. And I think for the future, we're really going to have to be able to understand big data. We're going to have to be able to do integrated analyses. And I'm going to tell you at the beginning of this talk, those of you that are not nurses, that in this work, nurses are the best phenotypers. I'll say that again throughout this talk. Nurses are the best phenotypers. And we really, really need to collaborate in transdisciplinary teams. I like this slide about sensors and smart homes. That is going to be the future. We're working with a group in the UK right now who are systems engineers, and they study traffic patterns and traffic lights in London. And they figure out, using analytic approaches, how to make the lights green or red to improve the traffic patterns. But they're also working with in-home sensors in geriatric patients, where they put a sensor on the refrigerator, a sensor on the patient's favorite chair, a sensor on the bed, a sensor on the staircase. And for the pilot study, based on patients' changes in movement and activity in their home, they diagnosed 6 urinary tract infections. We are going to have voluminous amounts of data. and then integrate all the rest of it. Genomics was going to be the answer. I actually don't think it is. I think it's going to be gene expression, methylation, the metabolome. We're going to need to get an integrated approach. But it all comes down to the individual patient. And that's what drives my research. So I'm going to take you back to about 1976. And I'd like to introduce you to Frank. Frank is a 65-year-old male who retired recently, who's an avid gardener, who enjoyed having a beautiful lawn, having a putting green in his backyard. Some of you know this story, who know me. His wife, Helen, noticed in May of '76 that he was becoming hoarse. He had an ENT consult. He was diagnosed with laryngeal cancer. He had radiation, underwent a radical head and neck. And in 1977, he recurred. He died. This is my father. Okay, that's me, my communion picture. My father died in intractable pain. And we did not know that he had neuropathic pain back in 1977. He changed my life, and I decided I would go back. And the one promise I made to myself, and you can correct me if I'm wrong at the end of this talk, I would never ask a research question that did not have clinical meaning. And that's really what drives our work at UCSF. So if you look at symptom research as a whole, most of the field has focused on studying a single symptom. And today that's pretty much true as well. Pretty appropriate to take a reductionistic approach. We often do that in science. Get the epidemiology of the symptom, the characteristics, and then the impact of the symptom on the patient. But I've never met a patient that has one symptom. Would you agree? I mean, in every field. So starting to think about this symptom experience, and we'll talk a little bit more about that in terms of this concept of studying multiple symptoms, studying how symptoms cluster together. So part of the questions, part of the things we've been trying to sort out is this dynamic nature of symptoms. And the idea that a single cross-sectional assessment, which is a good place to start, is not the only way to approach this field, and that we really need longitudinal data. And I'm going to show you some of our data as exemplars of that. The other issue, I think, in symptom science is what dimension of the symptom experience should we capture? Should we capture yes, no? Is the patient having the symptom? Should we capture severity? Should we capture distress? How much is this troubling the patient? And hopefully if I get to the network analyses, I'm going to try to convince you that those three dimensions are really, really different from each other. And that we'd be probably at this point in the trajectory of symptom science, we should measure at least those three dimensions. Trying to study how symptoms interact with each other is not a simple story. And I'm going to show you a little bit of the evidence by which we've been trying to tackle that, and then come back to this issue of inter-individual differences. So for those of you that are not in the area of cancer symptoms, I just put this slide up to show you kind of the prevalence rates for these, and look at how many approximately signs are. We still don't have really good epidemiological data. The ranges are pretty big. In our group, we study all of these things, and I thank God every day for patients that fill out our questionnaire booklets that are this big. Marilyn's smiling there in terms of the kind of work we've done. So you can see this is a huge problem. If I were to show you this slide in survivors of cancer, it's the same thing. And I bet this slide is accurate for most of the chronic conditions we see. Maybe some variability in terms of the specific disease, but these are pretty agnostic symptoms across chronic conditions. So here's the rub. The rub is this notion of variability. And those of us that have research careers know we try to get rid of it. We try to do controlled experiments, right? Where we set pre-specified inclusion, exclusion criteria. That is not clinical reality, all right? People vary, and I'm going to show you some evidence to support that idea. Why do I think it's really important to understand inter-individual variability? I have those bullets on the slide. We need to figure out who's at greatest risk. But equally important, and I'll come back to this point in a little bit later, we need to figure out who doesn't get symptoms? Who's got the protective phenotype, all right? I think as clinicians, we're worried about the people with higher risk, but we can learn a lot from people who have a protective phenotype. It's needed to figure out optimal treatment regimens, and in our work, particularly in terms of doing the biomarker work, we're really interested in mechanism. My doctorate's in physiology, so I really, really want to understand fundamental mechanisms that underlie symptoms. So now we're going to go in and I'm going to talk about some of the work that we're doing and how we've approached this topic. In terms of the single symptom of fatigue, we'll come back and talk about multiple co-occurring symptoms, and we'll see if we have enough time to talk a little bit about symptom clusters. So fatigue is a pretty ubiquitous symptom. The NIH is actually thinking about studying this across chronic conditions. There's been a fair amount of work done in cancer, largely because orthobiotech funded the oncology people to do this type of research. But it's got multiple causes. And probably one of the leading mechanisms by which fatigue occurs in people with cancer is these inflammatory mediators are released. They're called cytokines. And we've done some work trying to sort that. So here's an example of inter-individual variability. So let me explain this slide to you. This is a study of about, 587. The total sample for this study that I'll come back to a little bit later in the talk is over 1,300. But at the time we did this analysis, we had a little under 600 patients enrolled. They were all getting chemotherapy. And this will be a recurring model for some of the subsequent slides in this talk. We purposely did not enroll patients prior to their first dose because that would have been an impossible task. We get critiqued every time we put papers in that say, why didn't you enroll the patient the first time? Well, we couldn't do it. It was just technically too difficult. So the patients were enrolled either at their second or third cycle. Time 1 is prior to their next dose of chemo. So we speculated the patients would be recovered from their previous cycle. Time 2 was the week after they got chemo, so we thought acute symptoms. And then the third assessment was a week later. So theoretically, they're nadir. We repeated that for 2 cycles. So these are the mean evening fatigue scores of the patients in the study, pretty much what you would see in any journal article. Is that truth? Okay, here's truth. Okay. This is what's lovingly called a spaghetti plot, happily named, right? This is the truth in the sample. This is not this. So what does this mean? Who are these patients? And that's part of what we've been trying to understand. So typically, the approach that's taken to do this kind of work is analysis of variance, or if it's cross-sectional, you do a logistic or a linear regression to figure out prediction, right? Analysis of variance is a hard method for this because no data set has complete data, all right? And if you have an analysis of variance, you drop everybody to the lowest number in terms of all the people that completed the 6 assessments. So we've been using an approach, and I'd just like a show of hands. How many of you are familiar with hierarchical linear modeling? Okay, some of you. So let me just explain it for the non-statistician. It's regression over time. And the cool thing about it is you can have missing data in the dependent variable. So the patient can give you one assessment, and they're included in this type of modeling. It really, really allows you to begin to sort predictors of risk. So you set up the model, you figure out what the model is without any predictors in it, and then you go in and you can figure out what may be contributing to a higher or a lower trajectory of fatigue. I'm going to come back and do this latent class thing later. Okay, so in terms of this analysis, as I just said, you're doing regression over time. You can test different models. And because we were studying 2 cycles of chemotherapy, it actually turned out that it was a piecewise model. We studied 1 cycle and then another cycle in terms of predictors. You can choose, based on the literature, what types of predictors you want to test. In our work, we've typically looked at demographic, clinical, other symptoms perhaps playing a role in predicting the trajectory of fatigue in these individuals. And we've also put in genomic markers, and I'll show you a little bit about that data. And as I said before, you can have missing data on your dependent variable, in this case, fatigue. You can't have any missing data in the predictor function. That's the caveat with it. Okay, so back to this mess. How do we predict what's happening with these patients? Well, this is part of Faye Wright, who's in the audience. This is part of her dissertation research. She was kind enough to help collect data for this study with Marilyn Hammer and Fran Cartwright, then at NYU, now at Mount Sinai. But we had a theoretical list of predictors that was much longer than this. And Faye systematically did an analysis to see which demographic and symptom characteristics were associated with different trajectories of fatigue, as well as look at genetic pathways largely in inflammasomes, a subset of inflammatory biomarkers. And so I'm not going to go through the whole list. This work is published. I just want to show you a couple of examples of what we found. So it turned out in this data set across the trajectory of evening fatigue, if you were white, you were more likely to have higher fatigue severities, evening fatigue severity scores across the duration of chemotherapy. If you had, for those of you that are not cancer people, one of the things we use is a very standardized measure to evaluate functional status in people with cancer is what's called the Karnofsky Performance Status Score. It's basically a single item measure, goes from 0 to 100. 0 is dead, 100 is fully functional. We give this to patients to fill out. We don't put 0 on the questionnaire, okay? We start at about 30, which means they need to be hospitalized. And not unusual for this data, higher is better. Okay, so 100 is fully functional. People who had a lower functional status score reported worse fatigue across the trajectory. And this is what one of the biomarkers looked like. So this was a SNP in IL-12 beta. And if you were, let me just look at the slide. If you were homozygous for the minor allele, the CC allele, that appeared to be protective. You had lower levels of fatigue. And then we go in and we explain each of these in terms of the relevant literature. Okay, so here's the next part. So I want you to imagine being in a busy oncology, clinic, and you've got 100 patients. And to make the argument easy, they're all going to have breast cancer. So we'll take out gender. We'll give them all the same diagnosis, all right? And I know if I'm going to walk in and give those patients chemo, some of them will get the worst fatigue possible, high on the score. Some will get a moderate level of fatigue, and some will get very low levels of fatigue. So how am I going to figure that out in terms of this? And by the same token, I know that some patients will get very few symptoms. I know that other patients will get a moderate level of multiple symptoms, and a third group will get every symptom in the book with the worst severity possible. So those are the two questions we're going to try to address. And I'm going to start with fatigue first. All right? Again, approaching this from a very systematic perspective in terms of trying to understand phenotypic characteristics that predict these different groups, molecular characteristics, and then importantly, do these groups differ on patient-reported outcomes that we find meaningful? And the typical ones we study in oncology are function, cognitive status, quality of life, and survival. All right, so that's kind of the framework for this work. All right, back to the precision medicine slide. The idea with this work is to try to figure out, you've got the 100 patients in the oncology clinic, you want to begin to phenotype them and sort them by severity of symptoms with the ultimate goal of hopefully finding the best treatment for an individual patient. So the approach we've taken for this is what's called latent class analysis. How many people know about that? Oh, pretty good hands. All right, so latent means hidden. All right, so you've got all these symptom scores like the ones I showed you in that spaghetti plot. And this mathematical approach that's based on structural equation modeling allows you to group people who look similar to each other but different from other groups. All right? You can do this with cross-sectional data. You can do it with longitudinal data. I'm going to show you examples of both. The reason I like it compared to cluster analysis, there's hundreds of different ways to do cluster analysis. This approach, latent class analysis, has very distinct rules to say the optimal solution is 2 groups, 3 groups, 4 groups. All right? So it's really clear how you pick the groups. And I'm not going to go through the mathematics of this. But once you figure out the number of classes, each patient in your study gets assigned a class membership, and then you can begin to compare the groups or look at differences among the groups. So it's a pretty slick technique. All right, so to cut to the chase, all right, this is the same data for fatigue, evening fatigue, where we did a latent class analysis compared to the hierarchical approach I showed you. And you can see on this scale, about 4.3 is a significant level of evening fatigue, all right? So when the statistician did the latent class analysis, we got 3 groups. We got a group that pretty much through the course of 2 cycles of chemotherapy had relatively low levels of fatigue. Group that had moderate, or we claimed high because it was above the cutoff, and then a group that was very, very, very high. Would you like to guess how they differed in terms of demographic characteristics? Who's in the very high group? White? Okay. Men or women? Ah, okay. Young or old? I'm going to get some water. Little audience participation, never hurts. All right, so people are saying men. People are saying, how about younger or older? Okay, so here's the list. All right. I once did this for the UC donor board, and I got them all riled up about who's younger, who's older. We had a great time. So younger people, and I will tell you, in every symptom we study, younger people have a worse symptom profile. Now, we could spend time talking about that maybe in the Q&A. In this study, female gender, more symptoms, and that's true across many of our symptoms. It's not absolute, but many of our symptoms. Being white in this case, that's not always true. That's one we have to figure out a better measure in terms of social determinants of health. I'm not convinced we've got the right measures in that. We added, and for those of you that do this kind of research, asking about childcare, really, really important in terms of symptoms. So please add that to your list if you haven't. We asked about childcare and we asked about elder care. Those can be significant predictors. Not surprising, poor functional status, worse fatigue, that was confirmed with the HLM, and then worse comorbidity profile. This is one of the rare cases that cancer diagnosis mattered, all right? The chemo regimens don't matter, the severity of the chemo regimen, the time since diagnosis, stage of disease, none of that matters over and over again. This was one of the rare cases where, diagnosis mattered. The ones in red, as I noted on the slide, very common across many of the symptoms that we study. I'm not going to show you all the data, but we did do, as part of our molecular characterization, gene expression differences among the classes. And for this, we took the moderate and compared them to the very high to try to get that extreme phenotype approach. And what we found in this study were 29 different differentially expressed genes. But what's more interesting to me from a biological perspective is not the absolute genes that differ in terms of their expression patterns, but the pathways that we're uncovering, because that to me is more biologically relevant. And in this study, we found inflammation, immune function, carbohydrate metabolism, a pathway that had been, or a group of pathways that had been collectively defined previously as energy metabolism, but we were able to break them up into insulin metabolism, skeletal muscle injury, and then skeletal muscle energy, and then a new pathway that appeared called, that related to renal function. So we contributed, this was a very popular cartoon of some of the molecular mechanisms that had been implicated in fatigue that was authored by Leo Saligan down at the NIH, we added to that model and put in a few more pathways. So I think this is a systematic kind of stepwise approach to begin to understand this. This preliminary data requires replication. And I can tell you, Cord Kober, who did this analysis, just got a 2 percentile on an R01 to do a really deep dive into this data. So I'm really excited about that. So back to the question now. We're going to switch to multiple symptoms. You ready? All right, I said people don't experience one symptom, they experience multiple symptoms. And we've got our 100 patients in the clinic. All right, so it's the same study with different sample sizes as we progress through. And I'm going to talk about, particularly in this chemotherapy study, time 1, which is prior to the next dose of chemotherapy, and then time 2, which is the acute symptoms. So we're going to look at those 2 episodes in the first cycle that we studied. All right, so again, we did a latent class analysis, but this one is different than the one I described for fatigue. This is cross-sectional at time 1, and we gave the patients the Memorial Symptom Assessment Scale that was developed by Russ Portnoy at Memorial Sloan Kettering, right across the street, actually. All right, that instrument has 32 symptoms on it, and we said, okay, let's take the occurrence data from the patients at time 1 when they were theoretically recovered from their previous cycle of chemotherapy. And we were going to try to group patients on the number of symptoms they endorsed. Okay, so this is based on occurrence data. In order to make the mathematics work for the latent class analysis, we chose to include in the analysis only those symptoms that occurred in 40% of the patients, all right? Because if we had a lower frequency of occurrence, it wouldn't have been as robust a latent class analysis. So I said this instrument had 32 symptoms on it. We took those symptoms that occurred in only 40% of the patients. How many did we put in this latent class analysis? 26, pretty close. It was 25, okay, 25. So these are people who have recovered from chemotherapy, and they're reporting 25 symptoms, all right? We got 3 classes of individuals, all right, on those 25 symptoms. We got a group that was relatively low, and these are in descending order. You can see the black line is the mean occurrence rates. This was our low group. A moderate group that sat right on that mean across the occurrence rates, and then a high group. Okay, so pretty interesting data. The sample sizes, not insignificant, right? Not insignificant. So what's the next logical question? What do you think we ask next? How did they differ? All right, so here's the data on differences. I don't want to spend a whole lot of time on this. This is published data, but I want to point this out. Okay, look at the mean number of symptoms in these patients. So why did the lows have 6 and the highs have 20-some-odd symptoms? All right, and we can go through the other. Diagnosis didn't matter in terms of the distribution of patients. So the next question I had, is this a trait? I see some of you shaking your head, right? So if you're high going in at time 1, are you going to stay high at time 2? You want to take a bet on it? Huh? You're looking at me like crazy. Well, let me show you the data, all right? So is it a state or a trait? You know, did we just get lucky doing this? Is this a real phenomenon? Is it occurring over time? All right, so I did that already. Oh, so the way you answer that question is with what's called a latent transition analysis. So now you're looking at this latent class analysis over time. And you're basically starting to sort and see, do the people who started out high at time 1 stay high at time 2, or do they move around? That's the sum of all this, all right? So this is where prayer comes in, all right? So the rules are, you have to do time 1 first. And this is a bigger sample than I showed you originally. This is about 900 and some odd patients. All right, so you do time 1 again, and we got the same solution, basically. Then you do time 2, and you pray that you get 3 classes at time 2, all right? And guess what? We did. Notice the percentages changed a little bit in terms of the high, moderate, and low. But then, now that you've verified that at each time point you have 3 identified groups, you can do the combined analysis. All right? And let me jump to this slide, and then I'll go back. All right? So when you combine 3 groups at time 1 and 3 groups at time 2, there are 9 possibilities, right? There's the high, people who are high that switched to moderate, people that were high that switched to low, people who were moderate that went to high, people who were moderate that stayed moderate, and then all the others. The ones we were most interested in were the high, the moderate, and the low. And this is what their figure looked like. So is it a trait? Yeah, I think it's a trait. It's pretty cool, huh? Pretty cool. Pretty cool analysis, all right? Who were in the high class compared to the low class? Younger, female, childcare. In this one, because we had a bigger sample, we were able to isolate some specific comorbidities. Not surprising, but important as the population is aging, right? This data is all published, so you don't have to take copious notes. I'm watching you write down. Comorbidity and functional status. I know she is. Yeah, it's good to take notes. Trains the brain, right? It's pretty exciting data. All right, one more. Oh, just so you get an idea of the top symptoms, these are not inconsequential. Not inconsequential in terms of the severity of symptoms and what people are experiencing. Now, this is the last part. This is with the over 1,300 patients in this study. We did the analysis again. In this analysis, because we had more patients, and the more patients you add to a latent class analysis, and this is time 1 data, the more likely you are to find additional classes, all right? So when we did this, we got our relatively low group, we got our high group, but look at the 2 groups in between. The way that this slide is organized is with physical symptoms here, which essentially for the 2 groups in the middle overlap. And then look at the psych symptoms. Okay, we get a group that has very high psych symptoms, moderate physical. We have a group that has relatively low psych symptoms, but moderate physical symptoms. Now, the caveat, I'm not going to show you all the data. We have replicated this in a study in Australia. We have replicated this in a study in Norway. So this is real. I can't understand those 2 groups in the middle yet. We need to do more work. But I have to tell you, I have a Fulbright scholar from Hong Kong who's studying renal patients, getting hemo and peritoneal dialysis. He found the same groups. So there's something going on here, all right? Now, if I gave this talk next week, Tuesday we're meeting with, we got funded actually to try to do a genome-wide association study. And next Tuesday I'm going to get the results of that to see whether or not we could. It's a small sample. It will be a pilot with 1,300 people. But we're going to look at the extreme phenotypes, the highs and the lows, and see if we can come up with some differential marker. Now I'm going to tell you a clinical story because I've been in search of the markers for the worst phenotype, the high symptom burden. And our group has a firm belief that we have to go back to the clinics that we collected data at and share the story, all right? So we collect across the Bay Area, and I went back to one of the clinics in the North Bay, and I showed this data. I was sharing this story. And afterwards, a nurse came up to me and she said, "Chris, I am so happy to hear this presentation. And it was a lesson for me as an oncology nurse." And what she said to me was, "My mother and I were diagnosed with breast cancer at exactly the same time. My mother went through and had every symptom in the book with the worst intensity possible. I didn't have any symptoms. And for the past 5 years, I've believed they undertreated me and that my cancer was going to come back. What that said to me as a cancer nurse was, we do the chemo teach all the time. We have to tell people there's a range of symptoms that they can experience. Very, very, very important information. Really, really important. How are we doing for time? We're good? Okay. All right, let me say a couple of words about symptom clusters. We're changing gears a little bit. Maybe I'll stop a second. Does anybody have any questions on this before we? Yeah.
Depression.
Yeah.
What percentage of the patients were on antidepressants, and was there a correlation?
Very few.
So
very few.
I really wonder whether or not your data don't say that maybe you're under-treating depression in these patients.
Could very well be. Could very well be. I think many of them tend to be on more anti-anxiety medications than antidepressants, but the highs, yeah. Really, really important to look at. Very much important to look at. We have, and I need to go back and look at it. We actually have the Center for Epidemiological Studies depression scale on these patients, and we can look to see if the people in the two high groups have clinically. Yeah, but we actually have a full-bore measure of depression, so we can go back and look at that. How many? 7 minutes, okay. No, these patients filled out assessment books that were this big because the purpose of this study was actually to do symptom clusters. And so we got a lot of symptom data. Imagine getting 2 cycles of chemo and filling this out every week. A lot of data to try to sort this out. They were magnificent patients doing that. All right, so let me just show you briefly a little bit of work around symptom clusters. Our group has been interested in this. We were so bold as to write the first definition of symptom clusters that thankfully has been adapted over time. But really to try to understand how symptoms group together, perhaps in some meaningful clinical way. One of the underlying hypotheses of symptom clusters research is that symptoms that group together may share a common underlying mechanism, and we're trying to sort that out. There's probably some truth to that, but I'm not going to show that data today. So what are the ways to do an evaluation of symptom clusters? The early work was done using cluster analysis, which I told you is a pretty laborious technique that's got a lot of challenges with it. Most people now are using factor analysis. So the same kind of thing you would use for looking at subscales on an instrument to try to group symptoms that have a relationship with each other. We've been really, really interested, as well as a lot of other people who study this, to try to sort whether symptom clusters change over time, and do they change based on the dimension of the symptom experience that you're studying. So do you get different symptom clusters if you only look at occurrence versus severity versus distress? And really one of the unanswered questions that I believe the approach we're beginning to explore with network analysis may help us understand is, if you create symptom clusters, so you've got these groupings of symptoms, how do they relate to each other? Because they don't live in isolation as you would get with a factor analysis. So I'm just going to show you some data where we recently published looking at, from the chemotherapy study I just spoke about, symptom clusters in people with lung cancer. All right, and this is just for those of you that don't do it. We evaluated the symptoms on the Memorial Symptom Assessment Scale. We did exploratory factor analysis. This cluster analysis, or this symptom cluster analysis using exploratory factor analysis, was done on the symptom occurrence data 1 week after the administration of chemotherapy. And when you do the factor analysis, you get this list of symptoms that group together as isolated factors, and then we get to name them. You can disagree with those names, but that's what we named them for this paper. Just for those of you that are interested in this area, across most of symptom cluster research, you get a psychological cluster. That seems to persist, whatever diagnosis you look at, and even across chronic conditions outside of cancer. The others vary somewhat depending on diagnosis. So this is pretty nice, pretty clear factor analysis, a nice grouping of symptoms, but it doesn't tell you really much about how symptoms relate to each other. So as I mentioned, we've been working with this group at the University of Surrey trying to understand whether network analysis might help us understand how these symptoms group together as well as how these clusters relate to each other. And I'm pretty psyched about this work. So it's the same set of patients, except in this case, we use the total sample that I just described to you for the latent class analysis. And the question we asked in this study, if we apply network analysis and we try to create the networks using occurrence data, severity data, and distress data, do we get similar symptom clusters? And does the relationship among those clusters vary depending on the dimension of the symptom experience you use, occurrence, severity, or distress? Everybody with me? Okay. You get pretty slick pictures with this. All right? So for those of you that are interested in the method we used for this, we used an algorithm called the Ising-Fit model for the occurrence data. And then that's to create the original network. And then for the severity and distress data, we used polychoric correlation. So basically what you're doing, in my limited understanding of this, is you're creating big correlation matrices. And then the engineer who did this work for us, in order to get us to see the communities, the actual clustering of the data, he applied an algorithm called the walk trap algorithm on the networks to get the clusters. Okay, so this is the network with occurrence. All right, pretty interesting, huh? Not those columns by the factor analysis, certainly, right? The way you interpret this is the circles are called nodes. The size of the node is proportional, in this case, to the frequency with which the symptom occurred. So the bigger the node, the more likely that symptom occurred. And then the green and red lines are called edges. The thicker the edge, the stronger the correlation. 2 minutes. Okay, I'm doing okay here. We just got this published in Nature Scientific Reports, so you can go and look at it. So you can see these relationships make sense. If we take one of the simplest ones, that looks pretty cool, right? Shortness of breath, difficulty breathing. This is chest tightness. Sorry for the abbreviations off the symptoms. This is severity. So again, remember the question is, does the network and the relationship of the symptoms differ based on whether or not you're studying a different dimension? So what do you think? Little bit. Look at the stress. Way different, okay? And now let me apply the algorithm. So here's occurrence. Isn't that cool? This is when you put the algorithm on it to say, these are the symptoms that group together in clusters. So not only can you see, like an exploratory factor analysis where you got these isolated factors, you can begin to see where they group together. Pretty interesting data, right, to help us think about what's going on. So this is occurrence. This is severity, kind of different. And then this is distress. Now, one of the things I'm really interested in as a clinician, what's driving that network? Which symptom is driving the network? Yeah, okay. So in this type of analysis, that's not so easy to determine. All right, and in the 2 minutes I have left, I'm going to very briefly tell you, they use these fit indices that I don't quite understand. All right, so I'm going to cut to the chase. These are measures of association and strength of association. You want to guess the symptom that's driving this network? I was really bummed out, I have to tell you. It was not my symptom of choice. I was hoping for fatigue or something like that. Okay. Nausea. Nausea had the best relational indices based on that diagram I showed you. And what flowed from that was lack of appetite. Now, this type of network analysis does not demonstrate causality. All right? But there is one that does, which I'm learning a lot about. It's called Bayesian network analysis. And Bayesian network analyses has direction to the arrows. So even with a cross-sectional study, you can begin to see direction to the relationships. And if you do it over time with Bayesian network analysis, based on what the engineers tell me, you can theoretically see causal relationships. I can tell you we've just done this for these data with a Bayesian network. And what was most interesting to me is we looked at men versus women. The networks with Bayesian networks do not look the same. And in fact, the simplest way to describe that data, I don't have the pictures yet, they're still making them pretty. Women have a much denser symptom network than men. Interesting? And younger people have a much denser symptom network than older people. Pardon me? We haven't done that yet. I don't think we have enough. We haven't been able to cross gender by age. We're writing the grant to do that. All right? And we have longitudinal data, as I've showed you, to try to sort that out. So I hope I've inspired you to think a little bit differently about symptom science, and maybe these techniques can be used in your own lab. To me, this is going to be the future. All right? This is going to be the kind of integrated analyses we're going to have to do, which is going to take a lot of patience, a lot of patience. So maybe Rockefeller can build a collaborative for those, hint, 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 for those of us that want to study symptoms across chronic conditions, because I really think we have to do this across chronic illnesses. We have to, because I don't think the body makes 100 ways to make fatigue based on a specific chronic condition. The trigger may be different, but the underlying mechanisms, we're going to learn more from studying symptoms across chronic conditions. I'd be really remiss if I didn't thank all the people that I have the good fortune to work with at UCSF, at NYU, Mount Sinai, University of Pittsburgh. God bless all the physicians in our centers that help us. I've been fortunate to have a lot of funding from the American Cancer Society, as well as the National Cancer Institute. And I thank God every day for the patients that are willing to give us a sample of their blood and to fill out our questionnaires. Because without that, we wouldn't be able to do this work. And I see a lot of you nodding. And this is one of the bastions of places that recruit patients to studies. So I thank you for your kind attention. I'd be happy to answer any questions. Thank you.
Fabulous talk, and we do have time for some questions, so wait for someone with a microphone, because we want to make sure that our webcast people get to hear the questions as well. Wait a second or two. Here we have.
Yeah, please. Why don't we start here?
Right here. And
Pat, yeah.
Hi. Well, thank you. It's a really wonderful lecture. I'm really, thinking about these being traits is just so depressing. and then I wonder.
Why is it depressing?
Well, because then it's what can you do about it if it's a trait versus a state.
Mm-hmm.
And so I'm wondering if it maybe is, if we can look at it over a longer period of time, or to see if these are indeed traits that go across, different types of illness. Because, I don't know.
Yeah, I think this is one part of the equation. So one of the ways to think about traits is if you can identify people at higher risk, you can intervene, right? If we figure out a mechanism, let's say a deleterious mechanism or protective mechanism, that could be a potential target. But the other part of this story that I haven't told you about, and for those of you that are burgeoning PhD students or postdocs, one of the things I believe we need to study in people with chronic conditions is stress. Stress and coping. Because in our work, we've done measurements of stress in these patients, and we did a measure of perceived stress, so the stress they were experiencing at the moment we studied them. We did a measure of disease-specific stress, but the one I'm becoming much more interested in is cumulative life stress. And we did an assessment using a measure called the Life Stressor Checklist Revised. It's been used primarily in women who have experienced interpersonal violence. I am amazed at how many stressors these cancer patients had. And pretty horrific stressors, not just, a car accident, people being in jail, their own experience with jail, being sexually abused, being physically abused. We're writing a paper on that right now. And we don't have that on our radar screen with people. So I see this as a way to do risk profiling, but also to begin to think about mechanism and potential interventions. The people that are all low, they probably don't need our help. The people that are high, if we can figure out these Bayesian networks, maybe it's the psych symptoms, as you were saying, Barry, that are driving the network. And we have interventions for that. So be a little more optimistic. Yeah.
Please wait for the microphone.
Oh,
Crazy idea,
there's
but
no
I'm-
crazy idea.
But I'm full of them. So if the same person who you found risk for a certain symptom in one chronic illness got another chronic illness, and that had the same symptom, do you think the risk would be the same with a completely different chronic illness?
Yeah, I do. If we figure out the profiles, I do. And the reason I'm telling you that is because we've done some work with family caregivers of cancer patients. They are in some cases sicker than the patient, and they're taking care of the patient. So I think there's ubiquitous mechanisms for these symptoms, and that's a hypothesis. The challenge with answering that hypothesis is NIH is organized by diseases, okay? And the Nursing Institute, which would be interested in this as a topic, doesn't have enough money to fund these kind of studies. This symptom cluster study, would you like to guess the price tag for it? With your taxpayers' dollars? $8 million. $8 million. Now, half of that went to the University of California because we have a 55% indirect rate, but it's a pretty intensive study to recruit, have the nurses go in, assess the patients. Yeah, so the Nursing Institute, unfortunately, isn't going to pay that bill. That's why I'm on Rockefeller to do it. See, I'm coming back to that. See what happens when you invite me?
We're thrilled to
I know
have
he
you.
is. I know he is.
Wait,
Others.
we had some more questions. I want to make sure we
Yeah.
give everyone a chance.
Please.
Hi, this is so interesting to me because I work in an oncology clinic in the Bronx.
Yeah,
And
my old
your
stomping grounds.
And I see so many younger women with such levels of symptom distress, but such complicated histories of violence.
Yeah.
And it's something that we really need to identify and put the resources in early. It's not that they need, more opioids or more anti-anxiety meds. They need this. The histories of domestic violence is enormous.
They need coping strategies.
They need coping, yeah.
I can tell you, when we were doing this study, one of the IRBs required that we give the life stressor checklist with a clinician because they were worried somebody was going to jump off a bridge. And I walked in on one of the patients doing the interview, and I thanked her for being in our study. And she said, no, I want to thank you, she said. No one has ever asked me about these stressors, and I believe they've caused my cancer.
Yeah.
We have to begin to have that dialogue with patients. It's really, really critical. Thank you for
that.
You're welcome. Yeah.
Any other questions? Well, right over here, please.
Sure. Margaret, you always have a comment. That's good. You probably have more than one.
Who don't know me,
And
yes,
we're
I
all
always
friends,
do.
so I can say that.
Yeah, but the one thing that I was going to say is how many times have our patients said to us, I've had this stress, I've had this stress, I've had this stress. So it's not surprising. It's exciting in so many ways that you may be able to do some links because that's something that patients maybe feel in their gut, but we've never been able to measure before.
Right.
So.
Yeah. The other thing you should measure is resilience, because that is a really important trait. And we have some people in our study that were highly stressed, but they came out in our analysis as resilient. They had very, very high resilience scores. And we just finished this analysis, so I can't tell you much more about that than just, we had the stress people, we had the normative group, and then we had this resilient group that had higher stressors than the stress group, but they had the highest resilience scores of the whole cohort. And we're just starting to look at who those individuals are. So there's always questions to
I'll
ask.
just make, we have a question from
good.
from our webinar,
Very good.
I think.
Yes. Hi, Dr. Miaskowski. We have a question from one of our online viewers. Martha would like to know, was social support a predictor?
What's social support?
Was social support a
So we do a
predictor?
very bad job measuring that. The only two variables we use are lives alone and married partnered. Those are our proxies. And yes, living alone is generally associated with a higher symptom burden across many of the symptoms we study.
So I'd like to build on that. In the course of taking my training in opioid addiction
When did
problems,
you take it? Let me ask you. Because it's changed over the years.
Yeah, no, I took it as part of my license to get my license
Okay, you're in New York,
updated in New York State just last year.
Okay.
But the thing that struck me most was the predictor, the ability to identify a series of risk factors for people who get started postoperatively, where that combination of traits will be highly predictive of the likelihood of going on to opioid use disorder. But many of the things that were on that list look very much like things that have come out in your list. And I think it might be interesting to match them up
Yeah.
and see just how well they align, because I think this may be all part of one
syndrome.
underlying
Yeah.
issue.
Yeah, I've been talking to people about the transition from acute to chronic pain to addiction. And I think the phenotype between chronic pain to addiction is probably closer than we imagine. And can we sort that out in terms of a biosignature?
And certainly because of the public health implications right
absolutely.
now.
Yeah.
Oh, yeah, it's Marilyn. Last question. Marilyn
Okay,
Marilyn
Marilyn.
gets the last word, or at least the last question.
So I'm curious if I'm thinking about the Simonton and Simonton work years ago, personalities, A personalities, B personalities, suppression of cancer. Are any of your measures associated with personality testing?
So I hate personality, but the psychiatrist in the group wanted us to study it. And we have the NEO5, which is a very classic measure of personality. And we've done latent profile analysis on that. It's a pretty classic measure. Now I'm going to have to pull this out of the recesses of my brain. We had a group that had more neurotic tendencies. That's one of the subscales on the NEO5. They had a much higher symptom burden. So again, the whole psychological, as you were saying, prodrome and what that is and how we treat it and try to understand it.
Your question about resiliency is fascinating
Yeah.
because that would mix with the type A's and
Exactly.
you know, they're kind of
Yeah.
want
Yeah.
to overcome this.
So we need probably, I don't know, maybe 100,000 patients to put all the measures into a big predictive model over time to try to understand how these things influence each other. We've done a little bit of work with the group at Surrey trying to predict symptom severity at time 1. How well does it predict symptom severity at time 2? So if you got an oncology patient in the clinic who says, I have this much fatigue now, tell me how much I'm going to have a week later. We did it for depression, sleep disturbance, and anxiety. And we predicted almost 99% accurately what the score would be at the second measure. That's published in PLOS ONE, if you want to look at that predictive modeling data. So we have to follow that out further.
Well, please, thank you again.
Thank you. It was delightful.